Hi, friends. I'm John Kempf, hosting this podcast. I am passionate about developing regenerative agriculture systems that improve soil health, produce crops that are completely resistant to diseases and insects, and produce fruit of such an exceptional quality that we can have a legitimate conversation about growing food as medicine. I've discovered that there are many people with incredible knowledge and information about soil and plant systems and how to develop regenerative agricultural systems. However, much of this knowledge and this information is scattered. It's found all over the place. Some of it has been published in peer-reviewed publications, but there are many incredible stories and a lot of knowledge that has not been published and that hasn't been shared with many people. I started Advancing Eco-Agriculture in 2006 to bring this knowledge together in a more coherent fashion and incorporate it into products and growing systems that growers can easily put into practice. It's my personal mission to have these regenerative agricultural systems become adopted globally and become the mainstream, the status quo, against which all other growing systems are compared. To help achieve this goal, I want to share the knowledge that we have learned in the last decade and make it available to everyone. While we have developed products at AEA that embody the principles of regenerative agriculture systems and make them easier for growers to apply, this knowledge and these principles can be applied anywhere, and when they are applied properly, they will always increase farm profitability and resilience to climate stress. If you have any questions, suggestions, comments, or ideas, topics that you would like for me to discuss, please connect on LinkedIn or on Twitter, where my username is @VisionBuilder7, or you can also email me at john@johnkempf.com. I would very much like to hear from you and to hear your feedback. Be sure to subscribe to this podcast, and thank you very much for listening. Enjoy the show.
Greetings, friends, and welcome to the Regenerative Agriculture Podcast. On this podcast, we explore the science and practical application of regenerative agriculture ecosystems, how to regenerate soil health, crop health and quality, and increase overall farm profitability. Our goal is to give you clear and easy to understand information and actionable information that you can actually implement on your operation. In this episode, I'm very excited to welcome Bob Kremer to the podcast. Robert has a lifetime of very rich experience in the agricultural space, working both for the USDA and for the University of Missouri in a variety of different areas related to plant sciences and soil microbiology, looking at the interactions of soil microbiology and weeds, weed seeds, weed germination, and a whole range of different topics that tie into looking at soil health and developing suppressive soils. So, Robert, welcome to the podcast. We're very honored to have you.
Thank you, John. It's good to be here.
So, Robert, you've done a lot of very interesting work. What are some of your most memorable moments that led you to where you are today and some of the things that you worked on?
Oh, well, yeah, it's been several years to culminate to where I'm at today, I guess. But I think probably the first challenge was with the appointment with the USDA Agriculture Research Service when I was asked to look at possibilities of decomposing weed seeds in soil using microbiological approaches that really no one had really looked at doing before, because at the time that I came into the agency in the early 80s, it was pretty much everything was chemical based as far as weed management and actually any sort of pest management. And so to have a challenge like that was pretty unique at the time. And really, looking for native microorganisms that could essentially rot seeds in soil was was a pretty challenging task. But that really kind of led things into really looking at more at plant and soil microbial interactions, and leading to natural ways of trying to control weeds using biological control approaches, and that was kind of started the way into looking at what do pesticides do when they're in the management system, and that eventually led to my interest in soil quality, which was the big buzzword at the time, that evolved into soil health. And, with soil health, folks are looking at management assessment approaches. And at the time that we really got into it, that's when the genetically modified crop issue came into vogue or into wide practice. And then on top of that, you add the whole Roundup or glyphosate issue. And certainly no one was really looking at that as that being a factor that was affecting soil health. And so, we kind of diverged and looked into that for a while as well. So, I'd say I probably hit about a half a dozen highlights over the career that I've had. And those it always seemed like you turn the corner going to something else, and it was a very interesting challenge.
You've mentioned a number of very intriguing pieces that I want to follow up on, but let's perhaps begin at the beginning when you described your early work in seeking to develop microbiological control of weed seeds and weed germination. Did you have any success in developing biological controls? And are there any tools available for farmers to use today?
Well, I would say that a lot of the work that we started, a lot of the research that we started, eventually was served as a pattern for others to take that up and pursue that kind of development. We had some. My main task was to try to look at controlling these weeds in annual row cropping situations, and it was almost impossible because, especially in, if you want to call it conventional or modern type input-based agro-ecosystems, so many weed seeds had been built up over the years, plus the fact that they were so diverse. You had a number of weed seeds in the field that, although we found some bacteria and fungi that could affect maybe 2 or 3 weed species, we would still have, in some situations, 7 or 8 weed species in the field that would still be problematic. So it came to the point that in a Midwestern situation with corn and soybeans, it was almost impossible to come up with something that was effective biologically within the framework that the practices that were in place. And, I'm talking about a short rotation of corn and soybean, no cover crops to build up the microbial diversity, and continuous chemical application. So, but I will say that our work did kind of set the stage for others in other parts of the country that have developed these types of organisms. For instance, in the Pacific Northwest, where cereal crops are the dominant production system, and where there's only 1 or 2 weeds in the system, such as downy brome or jointed goat grass, they used this kind of approach, and there is some organisms available today that are under development for commercial application in those types of systems where it would work.
That is fascinating. I was going to ask you about other types of ecosystems where this would be appropriate. Are you aware of any development work that has been done that would be relevant for fruit and vegetable production systems, or where you have agriculture ecosystems that do have a longer crop rotation, incorporating cover crops and so forth? Has there been any work done on those areas?
Well, there's been a little bit. There's been some Canadian work. The Canadian Agri-Food Canada has really supported this type of approach. And for instance, there was a specific pathogen they developed for a particular weed in horticultural crops such as strawberries, and that was particularly effective. It was against I think it was an annual weed. It was a mallow weed, is in the cotton family. And there are a few other examples, but the really, the effort that has put into this relative to the number of products that are available is very lopsided. I would say there's not that much out there. And, I would say that one of the problems is for development of such a product to go to market. There's a lot of companies out there that don't want to take that risk, especially if if the crop is not a high value, that's not going to bring a lot of profit back to them. So that's always been an obstacle as well.
One of the pieces that you mentioned was the impact of pesticides on biological processes in the soil. Could you elaborate a little bit on what you've observed the impact of various pesticides to be?
Sure, it's been somewhat variable. What we're finding is some pesticides will be, let's say, detrimental or toxic to just a group of microorganisms in the overall soil community, and the same ones may stimulate other components of the microbial community, and then you would end up with this imbalance of the various microorganisms that are in the soil. Now, a very good example of this was in the late 70s, early 80s. We had a lot of soil-applied herbicides, fungicides, and insecticides, and these were going on at very high rates. Some of them, such as you might recall, Furadan as one of the insecticides, and EPTC or Sutan herbicides. They went on pounds per acre, and the other interesting issue with those were that their chemical structure had a group in there that was very similar to what you find in proteins, the peptide bond, or they called it the carbamate bond. And these really stimulated a lot of microorganisms that were able to use proteins. They just adapted and used those for metabolism. And the first thing you know, we had a lot of pesticide failures due to that. And then that's when we evolved into more post-emergence type herbicides that everyone thought, well, that's going to take care of that issue. And then what happened after that? We began getting a lot of herbicide-resistant weeds, and this was even before we had the Roundup-resistant weeds. We had a lot of the ALS resistance and even atrazine resistance in the weeds. What was also happening at that same time was that with continuous application of these same herbicides that was causing weed resistance, it was also altering the soil microbial community at the same time. Because what we often forget, let's say like in the case of glyphosate, is that even though we're applying it to take out all weeds in a genetically modified crop that is tolerant to glyphosate, only about 5% of what's applied is being taken up by the weed. We have the rest of it is taken up by the resistant plant, the resistant crop, and the remaining proportion is reaching the soil in some means or another. You add to that fact that being a systemic herbicide, that compound is released throughout the plant, and it goes to the growing points, which includes the roots. So you have a lot of that compound being released into the soil, into the rhizosphere, where you have the most active and dominant communities of microorganisms, because that's where all the nutrients are being released by plants, and that's where you find most of your microorganisms. So we get glyphosate in there, and that, I have to admit, over the last 20 years, that's the one herbicide I've worked with most, and we've documented that glyphosate will affect or is active in the root zone of genetically modified crops as just like it is in the root zones of weeds. It's released. It causes a lot of leakage of nutrients, the sugars, carbohydrates, and amino acids out of the roots, and this stimulates a certain segment of the community, and in this case, a lot of Fusarium fungi and some of the other detrimental bacteria like Agrobacterium and those that oxidize manganese that makes it unavailable to the plant. So often the resistant plant or the resistant crop, let's say it's soybean or corn, will not be infected by these organisms because they're able to resist it, but they are setting up a shift in the microbes in the soil that can be detrimental to other plants. It can be detrimental to other microorganisms. And now what we're finding is that, even though we've been told that glyphosate dissipates and is neutralized and is degraded, we're finding residual glyphosate in the soil now. And now, that's where we're at now. We don't know exactly what that residual glyphosate at whatever concentrations they are after the crop is gone, what they're doing to the whole soil ecosystem.
So this is fascinating. You've mentioned a number of interesting pieces. Let's perhaps begin with your last point of residual glyphosate. How long do you find that glyphosate residues are remaining in the soil profile after application?
Well, we've really started looking at this in a, well, not even a systematic manner, but at least in a cursory manner, beginning in 2014. And we were finding, and we were looking in field plots at the time, because one of the projects was, can we come up with some treatment that might be able to overcome the effects of glyphosate on the soil function or microbial activity and function. So we had these soils analyzed, and half the plots had not received glyphosate for at least a year, and the other half had received glyphosate during the growing season. And when we had that analyzed, we found that plots that had gone a year without Roundup application, some of those plots had as high residual glyphosate as the plots that received the glyphosate in the current year. So that was amazing to us, because that did show us, well, we are seeing a carryover of the glyphosate.
Wow.
And the other interesting thing is that it was random. We had how a field plot design is set up in a very structured arrangement. Maybe one plot that received glyphosate might be over 1,000 parts per billion, and then another plot that received the same treatment may only be at 350. So there's a lot of factors going on here that affects persistence of this particular active ingredient. And if I could just add one more thing. So that was the first year where we only detected glyphosate alone. In the last 2 years, we've been using a different lab that is able to separate glyphosate from its major breakdown product, which is aminomethyl phosphonic acid. That's the AMPA or AMPA that we talk a lot about. And that metabolite can be just as toxic as glyphosate itself. So we have those two compounds analyzed, the glyphosate and the AMPA. We find about anywhere from 10 to 50 times more AMPA than glyphosate, what that indicates is that glyphosate can be degraded by the microbes to one step. But then when it gets to AMPA, the enzymes to further break that down is not there within that microbial community. And thus, it's either they've been wiped out due to intensive farming or other herbicides or glyphosate itself, but it's pretty interesting. And we're finding the AMPA to be carried over by more than a year or two as well.
Well, what are the implications of high concentrations of AMPA in the soil profile? Do they have a similar effect on the soil biology as what you described for glyphosate?
Oh, yes. Yeah, it has a very similar effect, because the active group in glyphosate and in AMPA is the same. It's that phosphonate group. So as far as you listen to Dr. Huber and them talk about chelation of micronutrients by glyphosate. Well, AMPA will do the same thing, because it has not been degraded to the point where it loses that characteristic. So it can it can absorb the soil. It can exchange with phosphorus in the soil just like glyphosate.
So I have kind of two related questions. One, back at the beginning, when we first started this conversation, you mentioned that you had done some work looking at soil quality and looking at soil health. So I have a question around that, which is, what defines soil quality? What defines soil health? How do you know that you have a healthy soil? And then the follow-up question to that is, based on the pieces that we've just been talking about around glyphosate and AMPA, what are the long-term impacts of soil quality by these compounds?
Oh, okay. Yeah, so I'll go with the term soil health. Soil health is a little more holistic, and it really emphasizes soil biological function, and as well as the microbial component. Soil quality was originally proposed mainly to see how well your management is in terms of providing an acceptable agronomic production. you see, so and they didn't really emphasize the environmental effects as much. So anyway, that might be a picky point, but I'll just go with soil health. So what makes a soil healthy is, of course, you have to look at it in the context of the similar soils within a given landscape, and there are several indicators that will suggest good soil health, but the two main ones is soil organic matter or soil organic carbon and the microbial diversity. Certainly, we're getting to the point where we can quantify differences in soil health based on what I would call soil organic carbon quality, which is a fairly high proportion of the overall soil organic carbon or soil organic matter is in a pool called labile carbon or active carbon. And that's important because that is the portion of carbon that supports the microbial community in the soil. Plus, it can serve as a source of substrates or energy for growing plants, as well as the activity of the microorganisms that are involved in all the nutrient cycling and plant growth protection. So, having a soil with a good proportion of labile carbon is real important. Now, I can't tell you, what the proportion should be. Maybe it's 25 or up to upwards of 50%. That just kind of depends on the quality of organic matter that has been returned to the soil over the years. Certainly in a ecologically based farming system where you have a lot of diverse plant material and maybe compost additions and that, active carbon is going to be quite a bit high, and the quality is going to be as high as well. But then connected with that is this microbial diversity, and that primarily means that you have a diversity in the types of fungi and the types of bacteria, as well as a good component of things like protozoa that kind of complete the food web and feeding on these ongoing bacterial communities to kind of keep them in balance, but also recycles nitrogen, phosphorus through their decomposition of microbial biomass. Plus, the diversity of these, let's say the bacteria, is important because you need several different species in order to accomplish a function. In other words, an example would be if we have corn stover on the surface of the soil or just into the surface, one microorganism cannot break that vegetative material down to mineralize the carbon and nitrogen out of it and form organic matter. You have to have a successional group of microorganisms. Often we say they're consortia that work together to break all this material down and to get the beneficial effects out of that. And oftentimes we see this in soils that I would say are not as healthy. Soils that have been planted to, let's say, genetically modified corn, for example, modified for with BT production and maybe a Roundup ready or glyphosate resistance. Sometimes we find corn stalks in these fields for 3 or 4 years after the crop was harvested, and these things are just laying on the ground and not being decomposed. And I believe what's happening is that we have really caused an imbalance in the overall soil microbial community that just can't get the decomposition job done. So and there are other things you can check with for soil health assessments, such as rate of respiration or carbon dioxide evolution out of the soil, which indicates good microbial activity and enzyme activity and all that. And then, with biological indicators, there should be some physical and chemical indicators as well. And these are fairly obvious things that we've already known about through soil testing, such as, the amount of available phosphorus and pH, and then on the other hand, the bulk density, the aggregate stability or the proportion of aggregates in the soil. So, we take all those things together, and, I don't want to get into the technicality of it, but, we are coming to the point where we can model all these indicators, put it input these into a model and come up with a soil health index. And that's what's going to be important when we begin looking at, comparing management systems, as well as using that information to come up with guidelines that farmers can use to actually improve their management to improve soil health. And I think that's the most lacking piece of information right now, is that there's not a whole lot of guidelines out there for the guy who's out in the field. And we can research all this as much as we want, but it's not going to do any good until we have something that is usable by the farmer.
So if I'm a farmer today, how would you advise me to begin measuring some of these pieces? Are there any lab reports or any laboratories that are doing this type of analysis at the moment that I could begin establishing a baseline of where my soils are at the moment?
That's right. There are a few around the country that will do this, both commercial and extension type labs. There's a lab out in Nebraska called Ward's Lab. They do this kind of thing. They include a profile of your soil microbial communities. It tells you what the different main components are there, and gives you an idea of also how much microbial biomass you have. And Cornell University has a soil health lab that does similar things, and there's another one out east. And we have a soil health assessment center here at the University of Missouri. It's still kind of in the formative stages. It does real well with chemical analysis, and it does do one analysis for microbial diversity. So, one can send soil samples to any of those labs, and some have better reports on your results or interpretation than others, and that would be a good way to get started on assessing your soil health. Also, some states have incentives for farmers to do this. I know the Natural Resource and Conservation Service here in Missouri will subsidize some of the testing for farmers to have this done, because they have a program. I think NRCS nationwide has a soil health program where they're trying to improve soil health and maintain it, and they're trying to get farmers to get behind this, and they are paying them to do that kind of assessment.
Coming back to the conversation around glyphosate and AMPA, you've described a little bit what soil health means, and perhaps we have time to later explore the labile carbon, soluble carbon component or active carbon component. But I'd like to come back to the one question I asked, which was, how does glyphosate and the accumulation of glyphosate and AMPA over extended periods impact the overall soil health?
Well, I can give you a little bit of information that at least we can see that there's some indirect effect of continuous use of glyphosate on soil health, because we usually will measure a lot of the biological parameters when we set up assessments such as this. We see that there's some effects on some of the beneficial bacteria that are involved in plant growth promotion, such as producing plant growth regulators that stimulate root growth, and other beneficial bacteria that will produce pathogen suppressive compounds. And what we're noticing is that glyphosate tends to suppress those beneficial groups of bacteria. So that has an effect on subsequent plant growth as well. So we feel that there's a problem there. I briefly mentioned the effect on some of these microorganisms that are known to cause certain micronutrients to be immobilized and therefore not available for plant uptake. And one of these is manganese. And manganese is of course very important for activity of many enzymes that are involved in many of our metabolic pathways. And if that's tied up, you may have poor photosynthesis. You may have a poor amino acid formation, because you don't have enough of that micronutrient to satisfy the needs of the enzyme. So we have found, for example, the fusarium that will colonize the roots of plants that are treated with glyphosate. they are manganese oxidizers. So they immobilize manganese. And if they're on the root system, they're not going to be taken up. And if they are built up in the soil, whether there is a genetically modified crop there or not, they're going to remain in the soil and they're going to continue to immobilize manganese. And so if you don't have a lot of available manganese, that's going to affect the overall soil health as well. And then you get into a lot of other things such as glyphosate may exchange with phosphorus in the soil, and then you have problems with either excess phosphorus or phosphorus being, if it's not being taken up by the plants, it can be an environmental problem. So we mentioned the probability of effect of the quality of the organic matter, because we're using basically just two crops that are the source of the organic residues being returned to the soil. Certainly, if we don't have the microbes there to decompose them, or if there's not a diverse enough quantity of organic substances to help build up soil organic matter, that will affect soil health as well. Because, like I mentioned before, organic matter is one of the key indicators for good soil health.
In our experience working with many different farms, I would say, just off the top of my head, that greater than 90% of the farms that we work with experience severe manganese deficiencies. And I have wondered about the long-term effects. When you have glyphosate accumulation, you have this shift in the fusarium population, and you have oxidizing organisms that are immobilizing manganese. What are the long-term implications of that manganese immobility in the soil profile? How long does it take before that manganese might be released again and converted back into a form that the plants can actually utilize?
Yes, that is a concern, and that's I think there's several issues here. You have the effect on the shift in the balance of the microbial diversity and if that's shifted towards a lot of manganese oxidizers that is causing manganese not to be available. I think over time, if one were to alter the management to include, let's say, cover crops, or at least different crops within the rotations, and once you do that, you can stimulate other types of microorganisms that will help free that manganese, or at least compete with those oxidizers to reduce their impact on that. So that would be one possibility. The other, and how long that will take, it's hard to say. It may take a couple of seasons, or maybe less. That's something that really needs to be looked into. The other issue is something that we mentioned previously, is that how much manganese can be immobilized or chelated by the residual glyphosate and residual AMPA. That I think is a very serious issue, especially if we are in soils where the texture is such, or the level of phosphorus in the soil is low, that you don't have any competition with the glyphosate and AMPA. They will obviously chelate or immobilize manganese as well. I don't think we have any real good information on how long that can happen, or what the extent that situation is as far as tying up manganese over the long term. But I think taking all that together, with the shift in the microbes, the residual glyphosate and the AMPA, and the basically continuous corn-soybean rotation, if that continues, the manganese problem may persist.
When you speak of growing crops or bringing crops into a rotation to help reduce some of that manganese and increase its availability, what are some crops that are really effective at having a reducing effect and shifting the biology and the availability of manganese in the soil profile?
Well, I don't have any very specific ones, but certainly when you have a diversity of, let's say, cover crops in a mix, there will be some that will either support different microbial communities that are able to, let's say, mobilize these micronutrients, others that can actually plants that can mobilize the nutrients themselves. The kind of a common example is the use of, let's say, buckwheat or some of the brassica crops that can mobilize phosphorus or neutralize nitrates, those types of things. And then let's say you bring in something like sorghum into the rotations, grain sorghum or sweet sorghum, anything in that sorghum family. They, from my experience, they have a keen ability to host a lot of mycorrhizal fungi in the root system. And of course, the mycorrhizae are very adept at mobilizing many nutrients, not just phosphorus. And they would be, if you could put in a crop like that or other crops that can host mycorrhizae, that would be a very good way of maybe getting around the manganese problem and improving root growth, as well as improving the overall diversity of microbial community.
One of the pieces you mentioned earlier was the impact of genetically modified plants themselves outside of glyphosate and AMPA. How do GMOs impact the soil's microbial community?
Well, there's not a lot of information. We know that we found with soybean, for example, you have what happens during genetic modification. You can have some what's called pleiotropic effects, which are in other words indirect effects due to the genetic modification that is additional to the intended effect. In other words, in addition to the effect of making the plant resistant to glyphosate. And so there's things that can happen that we've noticed in the root system with some of the early genetically modified soybean varieties anyway, that even without being treated with glyphosate, the roots seem to release a lot more quantities of carbohydrates or soluble carbon and amino acids. And so this is problematic because what that does, it attracts a lot of microbes that readily use this material, and many of those can be potential pathogens. So you have a potential problem with not only some root pathology, but also it's possible to build up these segments of the microbial population that can be carried over from year to year. Another situation where we find these effects we noticed in corn, but not in all corn varieties, but in many varieties that had been genetically modified to be resistant to insects using the Bt gene. There would be a side effect where some of the corn stalks would have a lot more lignin in it than others. Lignin is very difficult to decompose. That's one of the reasons why sometimes we see a lot of that residue being carried over for 2 or 3 years in the field because so much lignin, it can't be decomposed very fast. And I think there are other situations that can occur. I mean, there may be some modification that we found. I had a Brazilian student here that looked at some of the nutrient composition, some of the omega fatty acid ratios were changed in soybean due to the genetic modification and that kind of thing. Now, I can't say for sure if that has changed with some of the more recent cultivars, because I haven't really been looking at that too closely over the last few years. But as you know, in our commodity agriculture, these varieties change almost from year to year, and some of the varieties that we were using, let's say 15 years ago, are not available anymore. So that's always another problem. You just don't know whether the effects of these newer varieties are any better or if they're any worse, unless somebody has a research program that's addressing that.
Your first point is very intriguing, though. In essence, what you're describing is that these crops and these plants may have the capacity to actually develop a disease-enhancing soil profile, which is interesting when you consider the long-term implications. Right.
Yeah, I mean, that was a complete unexpected result that we had. And we were comparing this to some of the old non-GMO varieties like Williams 82 or Maverick and these kinds of things, and they had much lower soluble carbon and amino acid release. So it was quite interesting to say the least.
What do you believe to be true about modern agriculture that many other people don't believe to be true?
Oh, Well, it's there are a lot of issues. If we can kind of go back to this whole organic matter issue. We're seeing a lot of soils under modern agriculture, and we say that we talked about the intensive use of chemical inputs and the GM crops and all that. We're seeing a depletion of soil organic matter content in these fields. I've been tracking some fields for 30 years or better, and soils that at one time had, let's say, 3% organic matter are now down to almost 1%. And I think a lot of folks will not, kind of dismiss that like it isn't happening. But when you use, intensive, let's say, nitrogen inputs like anhydrous ammonia and all that, and you're not putting in more organic residues, whether it's from the crop or with cover crops or organic amendments like manures and compost, that nitrogen has to be used. If it's not all used by the crop, the microbes are going to use it, and they are going to use whatever carbon's available in the soil. And I'm certain that's where a lot of that depletion in the organic matter is happening. And there are some real good studies over at the University of Illinois that have shown this, and even they were very much criticized by that. People didn't want to believe it, and of course, a lot of it, it comes down to money. It comes down to what the fertilizer companies are trying to make money at that. If you tell them that that some of their product is ruining soil, some of the soil properties, that's not going to make them very happy.
This idea of the soluble carbon component and the importance of the soluble carbon component has come up in conversation a number of times recently, and you've just expressed how we are losing that. How can we regenerate that in the soil profile?
Well, again, it all goes back to having plants, and I think I've seen this on your website. Having a diversity of plants on the soil and having living roots in the soil for as long during the year as possible. When you can do that, and harvest as much carbon dioxide and use the sunlight to produce the carbohydrates in the plant, and if you have a good profile that is inhabited by roots, you will get that soluble or labile carbon back. And the reason that is because many plants, well, I should say all plants, estimated depending on the plant, anywhere from 20 to maybe 50% of the carbon that is fixed through photosynthesis will be released through roots by the plant. And it's not because the plant is compromised like with a disease or anything. It naturally does that. And that carbon, supports, the microbial communities in the soil. So, I think something just as simple as mixing up that rotation. It's too bad that we don't grow as much wheat in the Midwest like we used to. That would be an excellent way of doing that. You follow corn or soybeans with wheat. You have wheat going into the winter. You can frost seed clovers or Lespedeza or whatever in with the wheat in the next spring, and you have a nice cover crop going into the next planting season. What better way to really get your soil revitalized than doing something like that?
Certainly, the farmers that we have observed who are developing strong cover crop systems, such as Gabe Brown and Dave Brandt and others, are regenerating their soil carbon and their soil organic matter very rapidly and producing some very nice crop results as well. So I think there's a lot of potential.
Yeah, exactly.
What is a book or resource that you recommend most often to growers to learn more information about any of these various systems that we've talked about?
Well, there are some around that are useful and are not that technical. The one on teaming with microbes is good. Yes, that one I think is a good overview of what's going on in the soil and describes what these different organisms are. I know he's got one called Teaming with Fungi, but I don't know if that's much more informative than just the first one that he wrote on teaming with microbes. Another one that I found that is very interesting is one called Mycorrhizal Planet. Are you familiar with that one?
I'm not familiar with that one. That's a new one.
Pardon me?
That is a new one for me.
Yeah. Well, you can get it from acres, and it's really good. If you wanted to focus on the mycorrhizae and what they are, what they do, and how to have those established in the soil, I think that's about as good as let's say at the high school level. I'd say, as far as understanding what's going on. I think it's that's a good one to have, and it's reasonably priced. So those are two that, from the standpoint of having a microbial emphasis is good. And then there are some others about, what is it, Save Our Soil or something like that would be good from a soil standpoint. I just don't have those titles at hand right now, but certainly I could provide you a list of what I have later.
I would appreciate that, and I will attach that list to the episode.
Okay.
What is the question you wish I would have asked?
Yeah, that's a good one. I'm sure this is something that I'm sure you have encountered. I have also, one of the things that I didn't mention of the many projects that I've had over the years is that I get asked to evaluate a lot of biological products, and now I don't know if a lot of the folks that you work with have looked into these kinds of things, but some of those are very interesting, and some of them I would not recommend at all. So that kind of question I thought maybe would come up.
So what of these various biological products, what are some of the ideas or the technologies that you're really excited about?
Okay. Well, there are some that I think. Okay, I think we got maybe back up a minute. There are a lot of different classifications of biological products, and I don't think anybody has really settled on anything that is set in stone, so to speak, at this point. But there are things like biostimulants that help with the microbial community activity, as well as the plant, especially in terms of root growth and plant growth regulation. There are things that are called probiotic type biological products, and these have the actual living organisms in them, and they are supposed to be selected that help promote growth of plants and maybe help some of the soil functions, such as cycling nitrogen and phosphorus and these kinds of things. Some claim to have nitrogen fixing bacteria in them, but I haven't had much luck in showing that actually happens. And then there's another class of products known as prebiotics, and there are quite a few of these out there. And what a prebiotic is a solution or a product that is made of the metabolites or the biochemical products of microorganisms, but do not contain living microorganisms. So you have that kind of thing, and they're supposed to again, they not only stimulate growth of the plant, because I've seen the chromatographic spectrum of one product, and it shows there could be 300 different compounds in this thing. They don't even know what they all are or what they all do, but they do know that some of them stimulate plant growth. Some of them are plant growth regulators like cytokinin and gibberellins and all that, and some of them stimulate the microbes in the soil. So here's the issue with all this, is that most of these products are, their effect or their efficacy is primarily based on, let's say, testimonials. They haven't really been tested that much by unbiased parties, let's say. And some, but there are a few out there that have been tested, like we've tested some, and some have been tested by other either institutions or companies to show some effect. So I guess what I'm saying is that those that I've looked at that work really well, I'm impressed with. Some of them have these specific plant growth regulators that really help with root growth. It helps with plant vigor, and we find that they will also stimulate certain microbial groups in the root zones or the rhizospheres of these crops. And of course, here's the other caveat to all this. A lot of these companies will approach us and say they want to test their chemical to see if they can offset the effects of glyphosate, because they know there might be an angle there that might make their product a little more valuable, to the farmer, because if they can say, well, glyphosate has these detrimental effects that have these side effects, but our product can counteract that. Well, that makes that product even more valuable. So, in some cases, we've been able to show that. Some of these products we tested for, more than 2 or 3 or 4 years, we consistently see that there is an offset. But the biggest issue I've had is that some of these products will do all that, will even actually help the soil health indicators and help plant growth. But then at the end of the day, at the end of the season, we don't see that much increase in yield. And I think that's the bottom line with these products, is that if you're going to sell these or buy these, you want to have a bit enough of a yield increase that will pay for the product. A few years ago when we had higher prices for corn, there are a lot of people using these things. Right now, not so much. But I do believe that some out there that are worth the money, because I've seen how they work. And I think that they not only help the crop, but I think they help offset some of the detrimental effects that has happened to the overall soil community due to some of the modern agricultural techniques that we're using.
We use we use a number of different biostimulants and probiotics, microbial inoculants in the crop programs that we put together. And as we have tested many different products from different companies over the years, one of the biggest challenges that we have observed is, and of course, we're looking at things on a field scale, and not always that they're not usually replicated to trials. But one of the challenges that we've observed is the inconsistency of results, where we might have a product that produces extraordinary results, perhaps, let's say, 30 or 40% of the time. So it works really well sometimes, but it doesn't work consistently. And that's been a very interesting observation, because it happens and has occurred with quite a number of products, which has led me to ask the question, or to wonder whether there is also an environmental component of what is happening in the soil profile, what's happening in that field, and do we really have the right conditions where we're giving an inoculant or a probiotic the opportunity for success?
No, I think you're right onto something there, John, because that's what I try to indicate to the manufacturers when we test these products here, because we're seeing the same deal. That's why I wouldn't make any conclusions on what we've seen on just 1 year data. We've got to have at least 2 or 3 years, and even at that, sometimes we do have this inconsistency in seeing positive yields. And the very thing that you mentioned about the other factors that enter into this, in as far as the performance of these products, that point is well taken. And I say that because we're located in a part of the state that doesn't have the best soils for producing corn and soybeans. I mean, it has this clay pan, and conditions have to be just perfect in order to get a very good, an acceptable harvest from year to year. So I think we're testing some of these products under conditions from the standpoint of soils that is pretty rigorous. And if we take this and do it, say, in some of the monosols, or the high organic or more fertile soils, I shouldn't say necessarily high organic, more fertile soils in the upper Midwest, we might see something completely different. So I think these companies, I know that they are not as flush as some of the big ag chemical companies, but if they really want to show how these products perform, they need to be doing this in different regions of the country, or even internationally, in order to really show how these products perform.
Absolutely. What is the one action that you would recommend that growers need to take?
Growers, from the standpoint of growers under any situation or any system.
Yeah, just in general.
In general. Well, here's the deal. I think we look at this some of the principles of sustainable agriculture. One of them is we got to keep the soil covered, and that doesn't necessarily mean that we have to have cover crops on it, but we got to have, after harvest, there needs to be that soil needs to be covered, because if it's bare, or if there's not much covered with the residue, it is exposed to the elements. There's hardly any restoration of moisture in there if it's bare. I think that is a key. And then, of course, keeping living plants on that soil is even better, even better yet, because we get that organic matter built up, it will absorb more soil. You won't have a lot of erosion, and infiltration will be more regulated. And I think now, so I think those are keys. If I had to say one thing to produce, there is one thing that if we have a little time, the one thing that we didn't mention, and these are things. This is a practice that we used to do, let's say 50, 40 years ago, was to integrate livestock in with the cropping system. I believe that if we could run livestock again out on the fields, and I know it's not going to happen, but maybe for family farmers it can, and we can have livestock out there that helps process the residues, or even graze cover crops and recycle some of the organic matter, stimulate the root growth by grazing. I think that's a great practice that we have pretty much forgotten about.
That's a very interesting comment, Robert, because that's come up in a number of conversations recently about the impact that livestock can have on rebuilding soil organic matter and kind of rebuilding the entire ecosystem to a much higher plateau, a much higher state of health and overall vitality and performance. So I guess the question that I would have for you is that there's been some discussion that perhaps the economics of beef production, if we were to manage our livestock, and more specifically, if we were to manage forages with the same level of intensity that we manage some of our other crops, the economics may actually be there to outcompete and to outperform current commodity crops, particularly with commodity crop prices such as corn prices being where they are right now. So do you have any insights to offer on that? What are the economic implications of managing grazing very closely and very carefully compared to current commodity crops?
Yeah, that's a good question. I don't have a whole lot of insight into that, but just knowing, in isolated situations where, well, your example of Gabe Brown is perfect for this, because they've got the system where they integrate their grazing herds into their forages that is then rotated eventually into row crops, as I understand it. So that the economics, I think, would be there. And of course, this is a bias. I believe the more exposure you have for livestock that are grazing, that are not confined, that meat quality is going to be so much better than what you find from a feeding floor. I think if one would push the pencil on that, I believe you would find that the overall profits in a system that would very much outweigh the way we are doing commodity crops currently. To me, commodity crops are just, it's just to emphasize a little bit too much. It's for the commodity market, for exports, for biofuels, and all that. Very little of it goes to feed, although a lot of soybean meal go to feed, but for livestock feed. But we've really gotten away from what an agro ecosystem should be. And I think when the family farmer or the smaller scale farmer had livestock integrated into the system, the profitability was there. It's just that it's been hijacked by industry. I mean, I think that's the bottom line. Now, if there were a way to kind of turn that corner and bring some of that back, I believe a lot of folks would be surprised how profitable that would be. But unfortunately, I can't give you any solid figures at this point.
That's okay. Robert, it's been a tremendous honor to have you here on the podcast and sharing your thoughts. Thank you very much. And we look forward to having you back at some point in the future. Thank you for sharing.
All right, John, it's been my pleasure, and I've enjoyed visiting with you. And I hope that this information will be useful to your listeners.
Thank you.
This podcast was brought to you by a great company that I work for, Advancing Eco Agriculture. the leader in regenerative agriculture since 2006. At AEA, we believe in challenging the status quo to find more profitable and regenerative ways to grow crops. We also believe that healthy plants are resistant to pests and disease, and that to grow healthy plants, we must first think differently about agriculture, about empowering life instead of suppressing life, about regeneration versus degeneration. To achieve this, we formulate and sell products that help growers produce higher quality yield with less risk of crop failure. In short, we help growers make more money with less risk. If you aren't yet an AEA customer, go to advancingecoag.com and use the code REGENERATE5 for 5% off your first order. Thank you for listening.